Dear student, today I am going to discuss a very important topic which requires a very clear conception to answer so many MCQs. That is the tubuloglomerular feedback and the autoregulation of glomerular filtration rate. As we all know that GFR, glomerular filtration rate, and renal blood flow, it is normally kept relatively constant despite changes in arterial pressure. That is, within a range of arterial pressure, change of pressure will not affect the GFR or renal blood flow. How it is possible? It is possible due to the intrinsic feedback mechanism that is present in the kidney. Why I am emphasizing on the word intrinsic? Because these mechanisms still functions if a blood perfused kidneys are removed from the body. If the kidney with its blood perfusion it is removed from the body, then also this feedback mechanism exists. So it is independent of systemic influences. That is why we can call it the intrinsic feedback mechanism of the kidney. And this mechanism actually maintains the autoregulation, and we can define the autoregulation by this term: the relative constancy of glomerular filtration rate and renal blood flow is referred to as autoregulation. There are many mechanisms of autoregulations. It may be a myogenic mechanism. Here in this slide or video, we will discuss about the tubuloglomerular feedback mechanism. Here in this diagram, we can see that a decrease in arterial pressure to as low as 75 millimeter of mercury and an increase up to as high as 160 millimeter of mercury, there will be change of GFR. It is less than 10%. It's almost constant. Okay, so in parallel with GFR, the renal blood flow is also auto-regulated, but GFR is more efficiently auto-regulated under certain conditions than the renal blood flow. Okay, but most important auto-regulation, we will see most efficient one is in the maintenance of GFR. In certain cases, there may be the renal blood flow is not that much well auto-regulated. At the expense of RBF, we can auto-regulate the GFR. That is different situation. But in most of the cases, GFR and renal blood flow, they are parallelly maintained within a range of blood pressure, say from 75 millimeter of mercury to 160 millimeter of mercury. Now the question is how it is possible. This is another very useful diagram that I took from Guyton, chapter number 77. You can see here. Here they have plotted the mean arterial pressure along the x-axis in the millimeter of mercury. In the above diagram, here they are showing the renal blood flow ml per minute, and here they are showing the glomerular filtration rate, that is GFR in ml per minute. So this red line, it indicates the glomerular filtration rate, and the blue line, it indicates the renal blood flow. Here we can see that if we increase the mean arterial blood pressure within this range, say it is from 50 to say 100, or in this range, we can see that renal blood flow, which is not 50, it is around 75 to 160, the renal blood flow and GFR, they are maintained in a constant level. And here one point is also important here in this diagram, that autoregulation of renal blood flow and GFR we can see here, but there is a lack of autoregulation of urine flow during changes in renal arterial pressure. Okay, although if the arterial pressure is changed within this limit, the renal blood flow and the GFR is auto-regulated, but if you can see the urine output, that is per ml per minute, it is gradually increasing. It is not kept constant. Okay, so this is a very useful figure from a textbook. Now we know what is auto-regulation. There are many mechanisms of autoregulations. It may be the myogenic theory, it may be the tubuloglomerular feedback theory. Okay, so by this autoregulation mechanism, within a range of blood pressure, GFR and RBF is kept relatively constant. We want to know the molecular basis of it. Now start with the condition when the GFR is low. When the GFR or glomerular filtration rate is low, normally the most common causes of low GFR is the less arterial pressure. That will reduce the glomerular hydrostatic pressure and that will cause the reduction in GFR. Okay, so by any reason, if the blood pressure is reduced and the glomerular hydrostatic pressure is less, so there will be reduction in GFR. So when the GFR is decreased, so this decreased GFR, it slows the flow rate of urine or filtrate through the loop of Henle. So this flow rate, that is the movement of filtrate through the loop of Henle, it is slowed down. Okay, so it gets adequate time. So that increases the reabsorption of sodium chloride through this ascending loop of Henle. Okay, so there will be increased reabsorption of the percentage of sodium and chloride ions delivered to the ascending loop of Henle, because we know that the descending loop of Henle, it is not permeable to any solutes. So when this slowed filtrate, it is coming to the ascending loop of Henle, as it is getting adequate time due to the slow movement of the filtrate, so more sodium chloride will be reabsorbed. When the filtrate will reach the end portion or the distal convoluted tubule at the proximal part, the concentration of NaCl will be less. Okay, so the concentration will be less at this specific portion, which is known as the macula densa. So the macula densa will get a less concentration of NaCl. So first is GFR low, second is reduced flow rate, third is more reabsorption of sodium chloride, and fourth is less concentration of NaCl at the macula densa. Okay, now this macula densa, why it is important? Anatomically, where it is located, that we all know, but just a brush up of macula densa. There are three types of cells in the juxtaglomerular apparatus. One is the juxtaglomerular cells, which are the part of the afferent arteriole. Another is the macula densa, which is a part of this green color cells, which is a part of the distal convoluted tubule. Another type of cell is the lattice cell. Okay, so this macula densa, so we'll see that when the thick ascending loop of Henle. So okay, from this picture we can see, so this is a glomerulus, this is a descending, this is a loop of Henle, descending limb, this is the ascending limb. Now ascending limb, when it is moving up, the thick ascending limb of the loop of Henle, it reaches the glomerulus. When it reaches near the glomerulus of the nephron, from which actually the tubule arrives and nestles between its afferent and efferent arterioles. You can see that this part of the ascending loop of Henle, and from where the distal convoluted tubule is starting, it is coming in a very closer to the afferent and the confluence of afferent and efferent arteriole. So this is the afferent and efferent arteriole at the confluence, when this ascending loop of Henle and the proximal part of the distal convoluted tubule, they are coming. So you can see at this specific area, there you will find some specialized cells. These specialized cells are known as the macula densa. Okay, so this is the distal convoluted tubule. It is at the confluence of afferent and efferent arteriole, and at this specific location we will find some specialized cells, and which is known as the macula densa, which is close to the efferent arteriole and particularly to the afferent arteriole. You can see here, this is the afferent arteriole. So particularly they are very close to this afferent arteriole. Okay, so this is the location of macula densa, and we know that macula densa is the sensor of what? It is the sensor of sodium chloride. So as we came to know that macula densa is actually senses the sodium chloride. So here I want to highlight one important point, that this specific autoregulation, that is the tubuloglomerular feedback, okay, it is used to perform, it is a kind of feedback mechanism that links the changes in the sodium chloride concentration at the macula densa with the control of renal arteriolar resistance. Okay, it is actually this feedback, it ensures a relatively constant delivery of sodium chloride to the distal tubule, and it helps prevent spurious fluctuations in the renal excretion that would otherwise occur. Okay, so the main function of this tubuloglomerular feedback or autoregulation is that it will try to stabilize, what? Now initial delivery to the distal tubule. Okay, so if this initial delivery is stabilized, then the output of sodium and chloride and other solutes through the urine, it can be controlled. So this feedback mechanism, it actually links the changes of sodium chloride concentration. We know that autoregulation of GFR and renal blood flow we are talking about. Now this GFR and renal blood flow autoregulation, it is linked by this sodium chloride concentration at the level of macula densa. So we know that when the GFR falls, so there will be slow movement of the filtrate. It will give adequate time for the filtrate to be undergo reabsorption. So more and more sodium chloride will be reabsorbed. Less amount of sodium chloride will be delivered at the level of macula densa, and it will reduce the activity of macula densa. So the decrease in NaCl concentration, it initiates a signal from the macula densa. Actually, this reduction will generate some kind of signal that has two effects in the renin angiotensin aldosterone pathway. Okay, so when this reduced sodium chloride is delivered to macula densa, two things will happen. One thing is that there will be increased efferent arteriolar resistance that will be mediated by the renin and angiotensin 2, and the second thing is there will be reduction of the resistance of blood flow to the afferent arteriolar. So afferent arteriolar dilatation and efferent arteriolar constriction. Constriction is mediated by this renin angiotensin 2 mechanism pathway. Now we want to know the molecular details of these two things. So when less sodium is delivered, so sodium depletion is there at the level of macula densa. So there will be up regulation of NNOS, that is a neuronal nitric oxide synthase, and the COX-2 or cyclooxygenase 2 in the macula densa cell. So this is the macula densa cell. When the sodium chloride delivery is less, so there will be activation of this nitric oxide synthase enzyme, and there will be activation of COX-2. So with the help of this COX-2, there will be formation of prostaglandins, and these prostaglandins will go to the adjacent juxtaglomerular cell. We have seen that these macula densa cells, they are very close proximity with these afferent arterioles, and on the wall of the afferent arteriole we will find these juxtaglomerular cells. Okay, so this prostaglandin that is synthesized in macula densa, it will go to the juxtaglomerular cells along with the catecholamines. These prostaglandin will enhance the renin synthesis and secretion. Okay, so they will stimulate the cyclic AMP production, and there will be release of renin from the juxtaglomerular cells. So this mechanism is clear. Less sodium chloride, Then within the cell there will be activation of N isoform of the nitric oxide synthase and the COX-2. These COX-2 will help in the production of prostaglandin that will go to the nearby juxtaglomerular cells, and along with the catecholamines they will cause the release of renin. So we can say that when there is decreased sodium chloride delivery to macula densa, that will cause the increased renin release. Now dilatation of afferent arterioles, how it is happening, we will come to the later part. So increased renin release we have seen. What is the function of renin that we all know? So we can see here, this is the macula densa cell and this green part is the JG cells. Okay, so within the JG cells you can see that this prostaglandin and the catecholamines, they are stimulating the CAMP, and there are so many intracellular mechanisms by which the renin is released. Now the function of renin, we know that the angiotensinogen that is released from liver, renin will convert it to angiotensin 1. Then angiotensin 1 will be converted to angiotensin 2 with the help of angiotensin converting enzyme, which is released from the pulmonary blood vessels, and this angiotensin 2 will act on the adrenal cortex. We know that there are 3 layers of adrenal cortex. So at the zona glomerulosa it will act to release the aldosterone. Okay, now these angiotensin 2, it is a potent vasoconstriction and it can cause the vasoconstriction of the efferent arterioles. Now angiotensin 2 is released. It preferentially constricts the efferent arterioles. So I want to emphasize on this term, preferentially. Okay, efferent arterioles are highly sensitive to angiotensin 2 and it constricts the efferent arterioles in most of the physiologic conditions. Okay, and we will see that if the efferent arterioles are constricted, that will increase the glomerular hydrostatic pressure, and in turn there will be increase in GFR. Okay, so now the one point is clear that there is constriction of efferent arterioles mediated by the renin angiotensin aldosterone axis. Okay, but the same angiotensin, if it is a vasoconstrictor, why it is not constricting the afferent arterioles? Now the afferent arterioles appear to be relatively protected from angiotensin 2 mediated constriction in most physiologic conditions. Okay, so how this protection is possible? It is due to the release of vasodilators, especially the nitric oxides. Okay, so we have seen that there will be activation of neuronal nitric oxide synthase. That neuronal nitric oxide synthase will release the nitric oxide, and these will actually counteract the vasoconstrictor effects of angiotensin 2 in these blood vessels. Okay, so although angiotensin 2 is released and there are receptors of angiotensin 2 is present in virtually all blood vessels of the kidney, the efferent arterioles are constricting, but the afferent arterioles are relatively protected from this constriction by virtue of this nitric oxide production within it. Now what is the net result of it? So this is the afferent arteriole, efferent arteriole, and this is the glomerulus. If the afferent is constricting, so obviously the glomerular hydrostatic pressure is reduced, so GFR and renal blood flow are reduced. If the afferent is dilated, then the glomerular hydrostatic pressure is raised, so both GFR and RBF are raised. Now when there is preferential constriction of this efferent arteriole, what will happen? The GFR will be raised due to rise in the glomerular hydrostatic pressure, but the renal blood flow will be reduced. Now when it happens, this constriction, when we know that when GFR normally falls, okay, and when GFR falls, when there is less blood pressure of less blood volume like this, okay, so although the renal blood flow is reduced here, that has an advantageous effect too. So as the renal blood flow is reduced due to the constriction of efferent arteriole, that will slow down the movement of filtrate through these peritubular capillaries. As it is slowed down, the movement is slowed down, more sodium and chloride, sodium or salt and water will be reabsorbed. So that will help in the to increase the blood volume. Okay, so this constriction of efferent arteriole, it will raise GFR, but it will reduce renal blood flow. But this renal blood flow will reduce the flow rate through this periglomerular capillaries, and there will be more reabsorptions of solutes, and that will help to restore the blood volume. Another important point which is related to the dilatation or reduction of vascular resistance of afferent arteriole is that one, we read about the nitric oxide synthase and generation of nitric oxide. But what happens is when there is less sodium chloride, which is entering the macula densa, the functioning of macula densa, some kind of signaling will be reduced. Okay, so if it is not reduced, what is the normal function of macula densa? So these sodium and chloride, you will see that they will enter into the macula densa cell via the sodium potassium 2 chloride co-transporter in their apical membranes. Okay. Now when normally the sodium chloride is delivered, sodium is entered within the cell, so that will enhance the function of sodium potassium ATPase, okay, which is present at the basolateral membrane, and there will be resultant increase in the ATP hydrolysis, which will cause more production of the adenosine. And adenosine is secreted presumably from the basal membrane of the cell. So this is basal membrane from where the adenosine is synthesized. Now this adenosine will act on the adenosine A1 receptor of the macula densa cells to increase the release of calcium from the vascular smooth muscles and in the afferent arterioles, and this will cause the afferent vasoconstriction and a resultant decrease in GFR. Okay, so this is, we can see here, there is release of calcium, and there will be calcium calmodulin complex, myosin light chain kinase, and there will be cross bridge formation and constriction of vascular smooth muscle. When the normal sodium chloride is delivered, it is not depleted, it is not reduced. Okay, so when the sodium chloride is high amount or in the normal amount is delivered, the macula densa will do what? It will release the adenosine, which will ultimately cause the constriction, vasoconstriction of the afferent arterioles. Now, when the sodium chloride depletion is there at the level of macula densa, this mechanism is prevented. It is blocked. So one reason is nitric oxide. They will cause the vasodilatation. They will protect the afferent arterioles. Another is this adenosine mediated vasoconstriction will also be absent in this sodium depleted condition when the GFR is low. So to summarize that, the decrease in sodium chloride concentration at the level of macula densa, it initiates a signal from the macula densa that has two effects, one in the RAS pathway that will increase the renin synthesis. How? Because there will be with the help of this COX-2, there will be prostaglandin synthesis. Prostaglandin and catecholamine, they will act on the JG cell and enhance the release of renin, and renin angiotensin 2 will be synthesized, that will cause the constriction of the efferent arteriole. Okay, that will try to raise the GFR. Another important thing is this nitric oxide will cause the vasodilatation of the afferent arteriole, and the dilatation of afferent arteriole is also mediated by non-functioning of this adenosine. Okay, so in a nutshell, there will be two, when the GFR is less, so GFR will be restored by causing two things. One is the constriction of efferent arteriole and dilatation of the afferent arteriole. Constriction of efferent arteriole is mediated by renin. Dilatation of afferent arteriole is mediated by nitric oxide and also by not functioning of this adenosine. This is another picture which showing when there is dilatation of the afferent arteriole and constriction of the efferent arteriole, the net result is increased GFR. GFR is raised. We started with when GFR is low, how this autoregulatory mechanism will restore the GFR. This restoration will be done by this dilatation of afferent arteriole and the constriction of efferent arteriole by this specific intrinsic feedback mechanism, that is tubuloglomerular feedback. This is a graphical representation of whatever we have learned. Thank you. And this is a reference, Guyton and Hall, textbook of physiology, chapter number 77, and the Ganong, it is chapter number 37.